The presentation today will focus on some fairly basic concepts around Sky0515, which is our lead compound for the treatment of Huntington's disease. It is a pill, a daily pill that is taken orally once a day, daily, and is designed to target the underlying cause of the disease with the hope of potentially being able to slow the progression of the disease. We are about to initiate our phase 2 study in Australia and New Zealand. This is the Falcon HD study, so you can see in our logo on the top here. This is the name of the study. It also has another name, which you'll see at the end, which is Sky0515-004-ANZ, but you don't have to remember that. Falcon HD is probably easier. And we'll also direct you to some resources for where you can find out more about that. But first, just to start off with, I want to talk a little bit about Skyhawk very briefly, and also Sky0515 as a molecule, and then we'll get a little bit into what's in the pipeline for us from the clinical standpoint. So just as a disclaimer, I am a clinical scientist at Skyhawk, and I'm a full-time employee at Skyhawk Therapeutics. Okay, so I just want to briefly talk to you about who Skyhawk Therapeutics is. Skyhawk Therapeutics is a biotech company that was started in 2017 with a headquarters in Boston, Massachusetts, and an office in Basel, Switzerland, as well as people located in various other locations now. And we're about 60 or so people. At the moment. The Skyhawk was founded by two co-founders who worked on a program for the development of a drug that is now in the market called risdiplam, which is for the treatment of spinal muscular atrophy, another rare disease. And this drug risdiplam parallels a lot what we're doing at Skyhawk Therapeutics, Huntington disease and for other indications. And this is a small molecule. Risdiplam is a small molecule that targets an RNA, and we'll talk a little bit more what an RNA is in the next slides, to modify the protein that comes out of that RNA in order to treat the underlying cause of the disease. And this is what Skyhawk's focus was founded off of. And the objective of Skyhawk is really to focus on this idea of being able to target RNA with small molecules in order to adjust proteins that are causing disease progression as small molecules or oral medications. And just briefly, but again, we have another slide to go into this. RNA is an intermediary between DNA and the production of protein. And proteins are often the biological components of cells that allow the cells to function properly. And these medicines that Skyhawk is developing target RNA to modify how it produces proteins and hopefully fixes the underlying cause of the disease and helps the cells function more properly. So as I've mentioned now a couple of times, Skyhawk Sky0515 is a small molecule and is taken orally by mouth once a day. So it's a relatively easy drug to administer. It has the potential, which we are hoping to show in ongoing clinical trials, to slow the progression of Huntington's disease by addressing the underlying causes of this disease. It works by ultimately reducing the level of harmful proteins that are linked to the disease biology. And furthermore, we have already started to conduct studies in humans in the clinic. And as of last year, we completed a healthy volunteer study up to 14 days, where we were able to demonstrate that Sky0515 was well tolerated and did not show any strong safety concerns at that point. Which allowed us to move into patients already last year, where we have a small study as part of our phase 1 initial study, where we're currently dosing patients. here in Australia. We have 4 sites, including Flinders and Adelaide, that are participating in this study, and that is moving forward now and will be up to a duration of 1 year. But what I want to talk about as we get further into this presentation is about another study that we're initiating this month, starting to recruit this month, which is our phase 2 study, and I'll give a bit more information on what that means towards the end of the presentation.
Sorry, do you need to?
That's okay. Okay, so now the next slides are going through some basics, just for your information. And this slide is really just to give a snapshot in general of what types of therapies are being developed for Huntington's disease right now. And it's really an amazing time in Huntington's disease because we, after many, many years of research, we understand a lot about the biology of the disease. And this has led to a lot of companies investing in development of potential therapeutics for Huntington's disease. We still have to get there to find, these disease-modifying therapies. There are none at this point, but I think we have high chances as a community, as a collective of many different research institutes and companies that are trying to find drugs that can modify this disease. In particular, many companies are going after the approach of Huntington lowering therapies, which is partly what Sky0515 is doing. And there are different ways that one can go about doing that, using different what we call modalities in terms of therapeutic modalities. And I just will focus first on the top because this is where Sky0515 applies, is being a small molecule, which is really a very small chemical compound that is manufactured as a pill. Very, very similar to what you take, if you're taking an ibuprofen or something like this. This is the same concept that you just take with a bit of water, and it just generally distributes quite easily throughout the body. It does have to be designed, and Sky0515 was designed to pass the blood-brain barrier to get into the brain, to get into the CNS. That is not always the case that drugs can do that, but Sky0515 was designed to reach the brain, which is very important in the case of Huntington's disease. Other modalities that are being developed you might have heard of are nucleic acids, or ASOs, antisense oligonucleotides, or biologics, and these can be antibodies or viral vectors in the case of gene therapies. And these are medium to large size molecules, and in this case, these therapies generally have to be injected closer to the. And so that means in the case of ASOs or nucleic acids and biologics, this just generally leads to an intrathecal or spinal injection. Now, these modalities also they stick around for a while, so that you don't have to do this every day, thankfully. But they stick around for a while, and that means that it takes quite some time before they're washed out of the system, which allows you to also administer them less frequently. Okay, so now I want to go through some fairly general concepts that you're going to hear a lot about, and I just want to make sure that you have a bit of information to orient yourself. So first, just basic questions: what is RNA? So RNA is essentially a messenger in the cell, and humans have DNA code, which is in every single cell, and this DNA code, at least part of the DNA, codes for thousands of different genes. And the way those genes work is they get copied from DNA into RNA, and RNA then uses that code, goes through some processing steps, and uses that to produce the protein. And it is the proteins that come out of the RNA that are the essential pieces of the cell for life and for having proper function of the cell, so that you have a healthy human being. So really, RNA is the intermediary between DNA and protein, and RNA is continuously being made in the cell when it is supposed to be. So there's a lot of checks and balances that the cells go through that define when an RNA is made, but it is continuously being made and degraded in a transient way, whereas DNA is there and does not go up and down. So the way we like to think about this, to make it a bit more simple, is you can think of DNA as a cookbook that has thousands of recipes in it, and those recipes are your genes. And RNA is when you take one of those recipes, so you open the cookbook, you have a page with one recipe, and you make a photocopy of that recipe. And that photocopy is your RNA. And you can take that RNA, and because it can transport itself around the cells, you can take that RNA or that one recipe and take it over to the stove where you want to cook your dish or make your protein. And that's essentially the process between DNA, RNA, and protein. Now, it does get a little bit more, well, that's the next slide. It gets a little bit more complicated. We'll get to that. But I wanted to also say, what does it mean when we say RNA targeting drugs? And that is that drugs that are RNA targeting drugs generally interact specifically with a specific RNA product that we're trying to modulate. And they often interact through binding to the RNA, and they will take the instructions that are normally read by the cell, and they'll modify that slightly. So they'll make a little edit to the RNA, and we'll see what that looks like in the next slides. And the result of this is that by using these RNA targeting drugs, we can change RNA function, meaning we can stop RNA from producing proteins, or we can modify the instructions so that it produces a different protein that is better for that person, or we can modify how stable the RNA is, how long it sticks around to produce proteins. Maybe it's going to stick around much longer because we want a lot more protein. Maybe it's going to go away much faster because we don't want a lot of protein. And ultimately, by modulating RNA, you have a means of regulating gene expression that is not permanent. So we're not permanently modifying the DNA code. And that means that if you stop taking the drug, that the cells will revert back to how they were behaving before or being processed before. So the last piece of this puzzle is a cellular mechanism called splicing. And this comes in terms of processing of RNA. So when RNA is first copied from DNA, it is copied into a fairly long stretch of nucleic acids, but not all of those are needed to produce the protein. And so what the cell needs to do is it needs to take out pieces of that pre-RNA and stitch together only the RNA that is necessary for coding for the protein. And that mechanism is called splicing. So this is essentially like editing your recipe. You can think of a recipe as you have your list of ingredients, and you also have in between that some instructions that tell you, how you want to use the list of ingredients, how do you want to prepare them, things that don't actually make up the dish, but tell you how to make up the dish. And you can take, if you cut those out and just put together the ingredients, then you have the exact code for the dish. And that's what happens with splicing. So when we summarize this and then see where Sky0515 comes into the picture, we have the DNA, which is your genetic code, which is in all your cells, and it stores the instructions permanently. That DNA gets copied, or what we call transcribed, into a pre-messenger RNA, pre-mRNA. And that pre-messenger RNA is this long sequence of RNA that needs to be modified first. And that's the splicing step. And the splicing step is where Sky0515 interferes. And it modifies the splicing step of the Huntington RNA, for example. And this will, as you'll see in the next slide, this will modify what the RNA does in the end. So after splicing, you end up with your mature messenger RNA, or final mRNA product, which is what is used for coding for the protein that helps provide the function in the cells. And what we want to do is remove the bad proteins in the cells that are causing the disease. So don't get too overwhelmed by this, but this is just a picture to indicate what happens without Sky0515, which is shown on top. And we're using HTT RNA here as the example. And so what you have here, you don't have to understand all this, but you have some boxes and lines, and the DNA gets the Huntington gene in the DNA gets copied into the Huntington pre-mRNA, which has all this sequence here. But again, not all of that sequence is needed to make the protein. And in fact, it is really only this box and the first box and the two blue boxes that need to be put together to make the protein. And so in this case, the lines get taken out and this pink box, and I'll talk about the pink box in a second. And so you get just these three boxes that come together, and then you get the production of your mutant Huntington protein, which is toxic to the cell and eventually forms these larger aggregates in the neurons and leads to the degenerative effects in the neurons. Now, when you introduce Sky0515, this is interacting at the site of the RNA where you have this pink box. And this is what we call a cryptic exon, which means that this is something that doesn't normally get included during splicing, but has the ability to be included. And Sky0515 emphasizes that ability. So it strengthens that ability to get included. And so in that scenario, you end up splicing out all the lines, but keeping the pink box. And the pink box carries a code that signals the cell to degrade the RNA very rapidly. And so what you get is, instead of having time to translate the code into a protein, you get the RNA just gets chewed up and basically goes to the waste bin. And so therefore you reduce the amount of mutant Huntington protein and ultimately the amount of toxic protein aggregates. So after many, many years of research, we're very confident that HD is caused by the mutation in one of the genes, one of the Huntington genes. And this is due to an inherited extended copy of a DNA sequence called the CAG repeat or CAG repeat, which is inherited on one allele of the HTT gene. With this extended copy of the CAG repeat, you get the production of the harmful mutant Huntington protein that then leads to damaging of the cells after long periods of time, and eventually to an effect that has a more brain-wide effect in terms of neurodegeneration that leads to the symptoms that we know of, such as movement disorders, cognitive impairment, and emotional disorders as well. But this is not the end of the story. There's a second piece to the puzzle that has come to light more recently. And that is that the CAG repeat that you're born with, when it's more than 40 or more repeats, you will get Huntington's disease. But as the time progresses, that CAG repeat, in particular in neurons, gets larger and larger. And that expansion over the lifetime of the patient is also what leads to the more rapid onset and progression of the disease. So not only if we remove the mutant Huntington protein, but if we could also reduce this expansion of the CAG repeat, we may further reduce or slow the progression of the disease. And without going into details, several scientists have put into a lot of effort into identifying what is leading to this expansion over the time in Huntington's patients. And they've identified a number of proteins that are facilitating that expansion. And there are two in particular that show to be especially important, and those are called MSH3 and PMS1. And PMS1 is a target that we were able to also incorporate into Sky0515 as a protein that we can modify to a lesser extent than Huntington, but that we can modify by reducing, again, very similarly to Huntington, reducing the levels of PMS1. And so our hope is that the combination of the mutant Huntington lowering and the PMS1 lowering will have a synergistic effect to not only deplete the cells of the toxic mutant Huntington protein and aggregates, but also to decrease the amount of expansion that is happening over time, which ultimately will slow the progression of the disease. So the end of that story is that HD is caused by mutant Huntington, but it is exacerbated by the continued expansion of the CAG repeats, which is in part facilitated by PMS1. And Sky0515 aims to reduce Huntington protein as well as PMS1 protein. And it does this by distributing throughout the body, including the brain, which is the most important region in the case of Huntington's disease. And this is what we are now in the clinic to try and determine if we can achieve this goal. So I have just one data slide for you here that I would like to slowly walk through, as I know this is not these types of slides are probably not familiar to you, but this was one of the results that came out of our first study in healthy volunteers, where we were able to look at how much Huntington RNA we could lower in blood. And this gives us an idea of how well is the drug working the way we expect it to be working, and to what degree we can see lowering depending on the dose. And so what you see here is essentially in this phase 1 study in healthy volunteers, this was run for 2 weeks, where the subjects took the drug once per day. And there are four different groups. One group is placebo, which means they are not on active drug. And you can see that there's not much movement in this group here on the left-hand side. The other 3 groups are on active drug at 3 different doses. So these are the blue bars, and those go from 1, 3 to 9 milligrams of drug. And what we have is a very nice dose-dependent lowering of Huntington RNA in the blood, ranging between about 20 to 70%. And the value of having this range of lowering of HTT RNA, which we expect to correlate to protein, and we will be measuring that in our continuing studies, is that we can now study and investigate what is actually the therapeutic level of Huntington lowering that is needed and safe. And so we feel like we have a very nice way to look at quite a large range to make sure that we are able to maximize, hopefully, the therapeutic benefit of this drug. So now just moving into the clinical development plan and our future study that will be starting soon. This slide is really just to present kind of a basic clinical development timeline. So you of course spend many years in the lab developing the drug, testing it on animals in cells, and making sure you have as much of the information you need before you go into humans. You then run a phase 1 study, which is typically in healthy volunteers and or patients. And this is a small study which is focused specifically on establishing safety of the molecule. And that's what we have completed in terms of the healthy volunteer portion. We expanded this study, this phase 1 study, to also include patients, which is currently ongoing, as I mentioned before, and that study will have a duration of about 1 year. We're now starting our phase 2 study, and a phase 2 study is a much larger study. Which is done to patient patients. And while it continues to address and focus on safety and make sure that we continue to see that the compound is safe, it also has the objective of demonstrating efficacy. And efficacy can be demonstrated through looking at two different markers. One called biomarkers, which are the physiological differences that happen when you have an effect on the body. So we can take blood samples and look at biomarkers such as HTT RNA to understand, or HTT protein to understand how the effect of Sky0515 is having on those markers. We can do brain imaging to see how the effect is on the brain volume, which we know over time in Huntington's disease, the brain volume decreases. So we would hope that we either stabilize or reduce the amount of decrease in the brain volume. But importantly, it also looks at clinical assessments, many of which you are probably familiar with, and these are the assessments that are often done in the clinic with the patient to look at motor impairment, cognitive impairment, and psychological impairment. And these are going to be some of the key readouts that we need to demonstrate efficacy. And by efficacy, I mean that to demonstrate that the drug works. The next stage will be a phase 3 study, and this is essentially phase 2, but much larger. And this is generally the study that takes you to the regulatory authorities hopefully for approval if everything is working well. So I've said this already, but Sky0515-004-ANC Falcon HD is a phase 2 study enrolling HD patients in Australia and New Zealand, and this is initiating now. Okay, I have just a couple more slides. I'll be fast. So the next 2 slides are dense with words, and I'm not going to go through all of this because you'll have handouts that will of all of this information. But I do want to just say that, unfortunately, clinical trials cannot be made available to everybody that has Huntington's disease. There's an important reason for this, and it does not mean that hopefully one day Sky0515 won't be available to everyone with Huntington's disease. But generally speaking, companies, when they design clinical trials, need to design trials in a way that allows them to demonstrate safety first and efficacy in as quick and as robust a way, as with a strong signal to allow the drug to become available to more people more quickly. And sometimes that means that we have to constrict the population that can participate in the study to allow for us to get to that point more quickly. And so I think one thing that is very important is what I say here, if you are interested in Falcon HD study, you should definitely talk to your primary physician or your neurologist in more detail, and don't go off of what I'm saying right now and decide whether or not you're eligible or ineligible, because it will be very basic what I'm saying right now. But there are particular inclusion criteria, and just to name a few of the most critical ones, you must be 25 or older at the time of consent, and you must have a genetically confirmed diagnosis of Huntington's disease with a CAG repeat of 40 or more. If you do not have this at this time, we can do this as part of the screening during enrollment. So this does not have to be done before. Then I think that the next part that is important to consider is that we do have several criteria coming out of the clinical assessments that are done that define what population of Huntington's patients we're looking for. And this, broadly speaking, applies to manifest HD in the early to mid stages of disease that we're focusing on. Again, I do want to emphasize this doesn't mean that this Sky0515 cannot apply someday to the broader population, and we will work as quickly as possible to make that happen. But that is that's, I would say, the most critical part there. And then there are other conditions, usually focused around other health concerns, or if you're on another trial, or pregnant, that would also disqualify you if from participating in this study. But again, this aspect needs to be discussed thoroughly with your healthcare provider, and not, don't take what I'm saying and make the decision from that just because it's not the full picture. So I'm sure that a lot of people here have questions about what it means to participate in a clinical trial, especially if you haven't participated in a clinical trial before. Again, you will receive this as a printout, and you will receive a lot more information from your physician on all of this. But just to highlight a few points, so why do we need to run clinical trials? Well, it is absolutely essential to run trials to make sure that we know that the new medicine is working, that it is effective, just as effective or more effective than current treatments that are available for the disease, if there are current treatments available for the disease. And if it has any side effects that might determine whether or not certain people can't use the drug or everyone can't use the drug. But these all center around demonstrating safety and efficacy to make sure that we have a good understanding of the molecule that we're going to more broadly distribute. Very important is that clinical trials around the world are highly regulated. They are regulated by laws, government laws, by ethical standards, and by codes of conduct that are independent of the company that is developing the drug. So that means that whenever a company wants to conduct a clinical trial, their protocol and other information is submitted to an independent ethical committee that must review this in its entirety and make their own judgment on whether or not this should go forward as a clinical trial. There can be multiple benefits from participating in a clinical trial. Of course, the main incentive is likely to potentially be on drug and have a therapeutic benefit from that clinical trial. But overall, participating in clinical trials advances the science and the understanding of the disease, and also allows you access to very medical experts and facilities with very strong knowledge and awareness of the disease that can help you work your way through the disease, but also learn more as you go through the trials. And then here it's really just going through what's going to happen before the clinical trial. And I talked about talking to your physician, getting all of the information, coming to the decision together with your physician. Is this the appropriate trial for you? You have always the say at the end of the day as a patient whether or not you want to participate in the study. During the clinical trial, in the case of Sky 0515, you will be taking the drug at home on a daily basis, and you will come in about every 3 months to perform some assessments. So it's not so frequently that it's entirely disruptive, but you will have about every 3 months a return trip to the clinic. And then, of course, after a clinical trial, this can basically proceed in 3 ways. One, that there's another phase clinical trial that needs to be conducted before potentially going for approval. The second would be the unfortunate situation where the trial did not demonstrate efficacy or safety, and then this would stop the trial and the research around this molecule. And the third is that at some point you have enough data and clarity on the efficacy of the drug and the safety of the drug that you're ready to go forward towards regulators and try to get approval for the drug. And the last 2 slides here are just and again, you will get this information, a list of the different centers that are participating in the study, the Falcon HD study, where the team is here to today and then over the week talking with a lot of HD people, HD advocates, clinicians, and participants, clinical sites that are participating in this study. And for more information, you can always go to clinicaltrials.gov using this identifier number, where you can find out more information on all of the aspects I was talking about in terms of inclusion, exclusion criteria, where are the sites, and so on and so forth. If you have any general questions or inquiries, you can email at this email address. And I think fairly soon, Skyhawk will have a web page that is dedicated to Falcon HD with more information there. So you can always look on Skyhawk, what is it, skyhawk.com?
Skyhawktx.com.
Skyhawktx.com to hope, I think, relatively soon get more information there. That's all. So thank you for your attention.
Thank you.
Well, thank you for coming to talk to us and explain all those details. We do try and explain when people come on.
I'm Karen Boundy. If you haven't met me, I'm a neurologist at Women's Medical Centre, and we've been doing a study.
Okay. Well, I'm only going to speak for about a minute. So, and so thank you for coming tonight to hear about the trial. And for those 200 people online, I hope they're able to appreciate the talk. And it is quite a complex area, dealing in anything with genetics, but I want you all to be reassured it's been done in very scientific manner and very rigorous research. And, we're hoping we'll have positive results. But as with any sort of research, you have to see how things pan out in the longer term. But we're, feeling very confident about this. So if you've got any questions about how to do a trial or what's been happening or about the scientific side, we'd be happy to answer those questions. And we've got our staff here in the front row who've been involved with the day-to-day running of the trial. So I'm sure they'll be happy to talk to you even informally later at the back of the room. Okay, thank you for coming.
We've obviously got questions. There'll be questions in the room, I'm sure. There are also some questions online. Sorry about me, for the people that are here have seen me going back and forth. For some reason, the slides were shadowing over it online, so I was trying to obviously fix that. So we had a bit of a technical hitch. So for everybody online, we apologise. We'll make sure that a copy of the slides is also available with the presentation if you want to have a look at them more closely afterwards as well. How about we start with a couple of questions in the room, and then Ali, are you there?
Sure am. I've got a list of over 20. So you start with a few in the room and then we'll go to a few online.
Okay, so anybody in the room got a question?
Yeah.
Yes, Phil.
So the protein that it's targeting, is it specific to the Huntington's protein, or is it kind of does it also affect others as well?
So it is affecting Huntington RNA, which is not allele specific. So we will be lowering both wild type and mutant Huntington. So then maybe that's part of your question.
I guess I was wondering whether there was any sort of
Other
unintended consequences of.
Yeah. So Sky 0515 was designed to have high specificity for Huntington. It also, I mentioned PMS1, so we do also lower PMS1 to a lesser degree. The key with small molecules is really establishing that window, that therapeutic window where you can target your target molecules, but not target other molecules, in this case RNA molecules. So if we were to go up in dose, then we would start having off-target effects. But in the range of dose that we're in now, we're really focused on the targets, the primary targets of our.
That was my other question, was about the dose. So when you went up to the 9 milligrams, I and it was quite a strong response to the dose.
Yeah,
So what?
Kathy,
so
can
was
I
that the
interrupt for
reason
1
that
second?
that you stopped there was because you were trying to keep them effect specifically on the protein you were targeting, or are you going to look at the high doses to see if there's some?
yeah, that's a good question. Actually, we don't think we should go higher because, again, we're targeting both wild type and mutant Huntington protein. We have a lot of, we have a clinical scientific board that is advising us on this, and they all believe that there is potentially, a good route to going to 70% lowering, but they would not go lower than that because we know that we need some wild type protein in the body to stay healthy as well. So that is the primary reason why we're not.
Okay, we'll take one more question from the room and then we'll do some online ones and we'll come back. So who else have a question?
Kathy, can I interrupt for a second, please?
Yes, sure.
Can you please repeat or get Megan to repeat the question just for clarity for anyone that's listening online, please?
Yeah.
Thank you.
Sorry, so the first question was, do we only reduce Huntington protein? And the second question was, why are we not going higher than 9 milligrams? Did you hear my responses? Is that?
Yes. Yeah.
Okay.
Thank you.
On top. Are they all given the medication or are some given at placebo?
Okay, so the question is, does everyone receive medication or are some on placebo, given a placebo pill? And the answer is there is a placebo arm. In the Falcon HD study, it's 3 doses and 1 placebo arm, so that there's 4 arms. Do we want to say what the ratio is?
Yeah,
Can you remind me of the ratio?
well, the dose level that we are using is 4 milligram, 6 milligram, and 9 milligram dose level, and placebo. So patients will be divided equally across 4 arms. So 3 out of 4 will get active from low dose to high dose. But we just don't know which one is the most effective one, the safest one. So that's what we're doing.
Okay, so just to repeat that, we have 4 arms in the clinical study. 3 are active. They're with 4, 6, or 9 milligrams of drug, and 1 arm is placebo. So you have 3 out of 4 chances of being on drug in the study.
Then you might go into an extension phase after that study.
Yes.
Everyone who's participated would have the opportunity to be on active drug.
Yes, exactly.
So there'll be a blinded phase where none of us know what goes wrong, and then you go into the extension, and then when that's completed, it's unblinded and you find out what you're taking for.
Yeah, so if you didn't hear that, so Dr. Boundy just clarified that the intent is to, after we complete the primary duration of the study, is to go into an extension study where everybody would be receiving dose.
Did you want to ask some of the questions you received?
Yeah, thank you. So there is quite a few of them. I'm not going to get to every single one, but we will. Hopefully, we've already answered some. The first one is around, people are quite interested to find out how many years do you feel it will be between now and phase 3, and therefore how long is. So I guess there's 2 parts to the question. How long do you think phase 2 and phase 3 will go for?
Okay, I'll start, and I might have Masood, maybe Masood can come up here if you.
Sure.
But the Falcon HD study, which is our phase 2 study, has a duration of 12 months. So it's a 1-year study. This, of course, enrollment happens sequentially. So this means that it will take longer than 12 months to actually complete the full study and get the data and analyze the data. And as we just mentioned, that might also get rolled into an extension study that will go longer. So we also are already planning our phase 3 study, and I'll let Masood speak briefly to that.
Right, so the plan is to have a much larger study in other countries. We are starting in Australia and New Zealand with 120 patients, and then we are expanding to other countries and go up to around 400 patients. So that's our plan. As we mentioned, it's going to have a double-blind placebo-controlled portion of 12 months, and then everybody after that will go into open label, what we call, which is, open label, and all patients will receive active. So we start slowly. I mean, right now, the phase 1 study that we are doing, patients are on placebo for only 3 months. Then everybody is rolling over the extension, which is happening right now, for another 12 months of receiving active. As we go along, we change the dose, the duration to get to a point that we are confident, how the drug works, the safety, and most importantly, the dose.
Masood, can I just ask from the back of that question, how long phase 2 will actually run for? You've said phase 1 is you did 3 months and now they're doing 12. How long is phase 2?
Phase 2 is 12 months of double-blind placebo control, what we call, that patients will receive either placebo or active. And then after that, everybody will roll into all active open label. And phase 3, the way we are planning it now is 18 months of double blind, and then roll over the active. But the duration, as Megan mentioned, includes a recruitment. So the whole phase 2 may take like 2.5, 3 years. But we are not going to wait for phase 2 to finish in order to start the phase 3. Phase 3 will start by the end of this year. So we are really hoping to shrink the duration of getting to an answer one way or the other. We are not going to do it sequentially, which will take forever. So we really hope to get to an answer in 2 to 3 years.
Thank you. Another question was around, whilst using or on the drug, HTT is hopefully being paused. Does that then mean that if a person stops the drug, they go to back to normal progression?
So it does. What it means is, if you stop using the drug, you will go back to the same levels of HTT production in the cells as before. Of course, there might be many reasons to stop taking the drug, and it can be a good thing to revert back to your previous levels if you're not responding well to the drug. But if, in the case that you've stopped taking the drug but are responding well, then you would start to go back to progressing. If Sky 0515 is effective at slowing progression and you stop taking the drug, then the expectation is that you would start, if you go off drug, you would start progressing again.
Yes, there could be some sort of reset that might happen if you have a period of time when you haven't progressed and accumulated more CAG repeats. You might still be better off than you were before.
Yeah, I mean, it's not that you've lost all. If it's an efficacious treatment, you will have gained some time in terms of having efficacy, and so you would have you might still have that effect for some time, but I think the biology tells us that eventually it would kick back in terms of disease progression.
Sure. This is a good thing about doesn't have any permanent change. So the important thing here is that you spend time on this drug, there is no permanent change, just effectively to be used.
Right.
So if you stop the drug, there is no change.
Yeah. So the statement was just made that it is a good thing that if you stop the drug, that you can go back to your previous biological state, because for many reasons it may be that it's not good for you, and that this would not be a permanent damage to your overall biology.
Kathy, do you have more questions on the floor? I've asked a couple. I can come back to more questions if you've not. I'm sure you've got questions.
And there's one here in the back. Go ahead.
I might have misread the chart, but I think that when you're going with the dosage, the first scenario, the 2% change with an increase, the rest were a decrease. That was.
Yeah. So the question is around the results that I showed as a bar graph. With the question was related to the placebo group, which was the far left, where it showed an average of 2% increase in the gray bar. So basically, this is within the range of variability of the assay. It's not statistically significant difference from zero change. Which you could see there were some lines that were going up, and that's kind of the variability. So when we measure these, when we develop assays to measure these biomarkers, we're not always 100% correct, and so you're always going to have a little bit of variability, hopefully within a very narrow range that we really optimize the assay to have that. And so in this case, 2% increase is from a statistical standpoint is equivalent to zero.
And that shows that placebo doesn't work. So we know it doesn't.
It's a good sign, because
that's good.
that was no drug. So we
Healthy volunteers.
And that's in healthy volunteers.
Yeah.
Right. So that's the reason that we use placebo, to show that our effect that we see in the active with 1 milligram and 3 milligram is real, because if you show exactly the same with placebo, we cannot say that it is a real effect. In that graph, you see that there is no effect in the placebo, but all other dose groups, they decrease proportional to the dose.
So you saw that quite good response to the dose at the higher levels. I mean, do you know if there's a correlation between reducing the protein then, and whether that sort of follows through to an actual reduction in the production of the mutant
Routine.
the mutant
Yeah.
protein?
So in the graphs, so the question is around if I understood correctly, this is related to the graph that I showed, which was showing the reduction of the RNA in blood. And the question is this correlated to the reduction of the protein? And we don't have the answer to that yet, based on in human, yes. Thank you. In animal species and in cells, we do see a 1-to-1 correlation between this. So this is what, and in other clinical trials that are focused on protein lowering through RNA mechanisms, we've also seen a similar amount between RNA and protein. But we have yet to demonstrate that, and we are in the process of doing that now. But the expectation is that yes, it will correlate to the protein reduction.
Okay, Alison, back to you.
Thank you. Thanks. I've had someone ask about the process of seeing how do they start finding out information about it and seeing if they're available. And I know that there was a slide before of where all of your sites are going to be. So there are 10 sites across Australia and New Zealand for phase 2. So it's a two-part question. I've got someone in Tasmania that is wanting to know if they would be able to participate because they're not in any of those states that are listed. And 2, how do people actually see if they're eligible? Who do they go to?
I can take that question. For the eligibility, as Megan mentioned, we really need you to talk to your physicians and take it from there. These sites, when they get activated, will have their activation status on clinicaltrials.gov. And if you talk to them, directly, they can tell you exactly when they are going to be activated. For patients in areas where we don't have a presence, primarily we do everything we can to give a chance to those patients to participate. And specifically for Tasmania, we've talked with Calgary, with Dr. Lee and others to see if there is a way for those patients to actually travel to Calgary to participate. We have a patient assistance program where we can pay for flights, accommodations, and so forth. That's all we can promise at this point. We are continuing to assess whether we can have a site in Tasmania. And if that happens, we will open up. But in the meantime, that's the solution that we have. For people outside these catchment areas, we'll provide these assistance to participate.
For the potential participants to note that just because you're not in one of the primary areas, it doesn't mean that you're excluded. So please still reach out.
Exactly.
For example, we have patients come from our Gambia that are willing to be in the study.
Right.
Fast.
So yes, we can. We will do everything we can to make that happen. So please reach out to the nearest or any sites who are active, and we'll do what it takes.
Thank you. One more question around how many patients are you looking for phase 3? And if a person is not in phase 2, will they be eligible, or is there an option for them to still participate in phase 3?
Well, for now, we are focusing on phase 2. We really don't use phase 2 and phase 3, very literally here. We have one program and we enroll patients as we go. Right now, we are assuming 120 patients to be recruited in New Zealand and Australia. If we reach that number quickly, we may decide to extend that study and go beyond 120. Whether or not we change the exclusion criteria for future studies, we just don't know yet. I mean, at this time, this is our best estimate, best guess that this patient population that we have defined is going to be the most suitable. Whether the next study would be different, we just don't know yet, unfortunately.
Maybe just to add one point to that, emphasizing what Masood said, is that what we're calling phase 2 and phase 3, they're actually very similar, almost essentially the same trial design, just run in different locations. And we're expecting to use the data from both these studies. So the study that is now starting in Australia and New Zealand, and the broader worldwide study together to use both those sets of data for towards getting approval of the drug.
Very good questions.
Let me just repeat the question.
Oh,
So the question is, how do we come to the number 120 for a trial?
Right. So when we start, we always start small. So our phase 1 is only 24 patients, which is happening now, and it's moving forward. In phase 2, we look at what is feasible. So we did a lot of studies across multiple sites. We thought that enrolling 120 patients makes sense. And the next question is, what is the basic question that you're asking in this study? In this study, the basic question is whether 4 milligram, 6 milligram, or 9 milligram is the right dose in terms of reducing the Huntington protein and potentially resulting in good clinical outcome. So that's the basic question at this stage. When we move into what we call phase 3 or larger study, which we are planning to do with 400 patients, at that stage, we actually do statistics around clinical outcome and see, for example, if we want to improve chorea, how many patients we need per dose compared to placebo to show a statistical difference between the two. And that's how we get to these numbers. So it's a combination of what's feasible and what's scientifically appropriate. Alison, back to you.
Well, I might just ask one last question, Masood, and then maybe you can check with the floor again. I think quite a lot of our questions have been answered, not all of them, but quite a lot. Is it possible to be enrolled in multiple trials or to be part of multiple trials at the same time?
No.
No, unfortunately not. So in our study, we can only accept patients who are not part of other studies at this time. If they are, for example, part of a study who are just looking at natural history of the disease with no drugs, that's acceptable. Yeah. But otherwise, they cannot be part of two what we call investigational drugs because we just don't know which one is causing.
Yeah, so
what.
for example, you could be in the MAP-HD registry, because that's a registry, but
Exactly.
you could be in a clinical drug trial, that's a very different things.
Right. So if you are receiving an investigational drug as part of another trial, you cannot be part of our study.
Because how would you know which one?
Exactly, which one is what?
That's obviously decided.
Right.
So.
Yeah.
That makes sense. Thank you. Maybe check if you've got any more on the floor there, and then we can see from if we've still got some time left.
Very good questions. Thank you.
Anybody else got questions through? Or like, is it really big answer? People there any questions? Yes.
So, I've already pushed here. As a issue, I want to know, as what about people who haven't got a lot, families are in French. A lot of some dead kids. Europe.
Darryl was just asking, more about his family that have got COVID, and whether they can apply to do it.
Yeah.
So the question was, the gentleman wanted to know if his family members have the positive gene, if they can also apply for the trial.
So potentially, yes, they need to speak to a physician. If they haven't been diagnosed yet, then they have to go through the diagnostic process and understanding if they're at the right stage of progression of the disease for this study.
Any more questions in this room? I think we're done. Ali, what about you?
I'm trying to field some of the questions online myself as they come through. I have been asked again, will you cease all other medication whilst you're on in the study. So HD related or not HD related. Can you just clarify that, please?
Yes, if you are receiving treatment for your HD and you are stable on that, and as long as your physician has prescribed it and it's not part of a clinical trial, you can continue receiving those medications throughout the study. What we want to avoid is to start a new medication once you come to the study or change the dosage significantly, so we don't know exactly what the effects of our drug is versus other medications. But for example, if you are on antidepressants or if you are on other drugs that are prescribed by your physicians for your HD, you can continue receiving those. You do not have to stop taking those medications. But again, it's very important not to start a new medication during the study.
Well, if you're going to start, you want a period where you become stable
Right.
before you then use the trial.
Right. So if you need to start a new medication, we may have to decide to stop 0515 and discontinue from the study.
Or
Unfortunately,
you spend a bit of time getting your new drug established and everything settled and stable before you then anticipate entering the trial.
right. Exactly. So if you need to start a medication now, we require that you need to be on that medication for 12 weeks. So everything is stable, and then you can come to our study and continue that medication.
I think I'm just going through some of my questions. Kathy, I don't know if you can see them as well, just to check that there's anything that's really standing out that we've missed.
No, I did have a quick look before, but I'm sure more have rolled in since I've come out from behind my computer.
Sorry, there's another question come through about maximum age for the trial, but and the conditions or the requirements for the trial, but they are explained, or they were explained in a slideshow, and we've got those handouts as well. Unless you wanted to say anything further on that, Masood or Megan?
We don't have a maximum age, as long as they are older than 25 years old, they should be 25 or older. They should be eligible and they meet all the inclusion and exclusion criteria. So we don't have a maximum age per se. And all of these informations are provided in booklets that we have here. And Alison, you have access to that you can distribute.
Yeah.
We've received the HREC approval for these pamphlets to be distributed across patients for their information, and we'll come up with more materials. Basically educational. The next step will be some flyer that explains what happens during this study, like why we do MRI, and why we need to do lumbar puncture, and so forth. So we will continue to provide educational material.
Okay. So just from my end, a couple of things. There's some questions around when people will get the recording. We will aim to do it within a week, but I'm not going to promise it will be done this week because it takes a little bit of time, and we will also send copies of all of the slides that Skyhawk have put forward today as well. One last question, if I might, if that's okay. I've been asked, have the FDA or TGA given any fast track or orphan drug designations?
Great question. We are actually going to work with TGA towards the end of this year and request a fast track. and we'll keep the community informed.
Thank you, Masood. I think I've answered the majority of questions. I'm really sorry if I haven't gotten to anyone. It's certainly not intentional, but trying to manage the questions is a little bit tricky. So from here, sorry, Masood.
No, I just want to say thank you, Alison.
So
All the way here.
Oh, sure. I've been here, many times. We'll keep coming back. We'll be back, definitely.
Thank you to everybody, both online and also in person. I'm sure there's lots of conversations still in person that will take place in the SA office there. As I said to you, we will certainly produce the recording and also all of the materials and send them out via an email to all of the participants who have registered both online and in person. We will also have them available on our website or a link to somewhere where you can download them as well. And like I said, I will try and do that within the week, but let's try for the end of next week at the very latest. We'll see how we go. Thank you to Skyhawk for actually wanting to come out and, put this presentation on. The community is really excited about the options for clinical trials. And I think tonight's presentation was extremely easy to, well, I shouldn't say extremely easy, but easy to follow and has certainly given some hope. So from here, if anyone does have any questions, just a reminder that all of the sites, of which there are 10 of them, will have contact details for you to be able to get in contact with them. And like Skyhawk have just alluded to, they would rather someone come and ask about a trial than not be on a trial. So if that means that you're not really close to a location and you think that you might be eligible, you can certainly reach out to any of those sites that are there, but we will have those details in an email as well for you.